This next session I know is something everybody's really interested in, and that's really, I think we call it a bird's eye view of what's going on with ataxia research. And nobody better to make this presentation than our Chief Scientific Officer and my colleague. Please welcome Dr. Lauren Moore. Lauren, hey. Thank you.
All right. Good afternoon, everyone. Can you hear me okay? So I'm Lauren Moore. I'm the Chief Scientific Officer at the National Ataxia Foundation, and it's really surreal for me to be here. The last time I stood on this stage was in 2016. I was just a grad student. I think my boss put a good word in for me and got me an invite and let me share a little bit about my research back at the 2016 AAC. So it's pretty astounding that I get to come back here and speak to you all now as the Chief Scientific Officer. So I've no financial relationships to disclose. As I mentioned, I'm the CSO at NAF. What does that mean? So in this role, I oversee, along with my really wonderful research team, a number of different programs and initiatives. They're all a little bit different. They're really wide-ranging. But overall, the goal of all of these programs is to try to move research towards the development of effective treatments for ataxia. That's really the end goal. That's what we keep in mind every day. And so first and foremost, what we do is we invest in research. One of the biggest investments that we make in ataxia research is through our annual grant program. We give close to about $1 million a year in annual research grants. And I think you would be pretty hard-pressed to find any ataxia researcher in the world right now who hasn't either directly received an NAF research grant or has been a participant in a project that was partially funded through NAF research grants. These grants are small but mighty, just like the National Ataxia Foundation. What they really allow researchers to do is to take big risks. And these big risks can have really, really big and important outcomes and can lead to much more funding being brought into ataxia research. My research team has grown a lot over the last couple of years, and what that has enabled us to do is really focus on growing the resources to help educate and engage and empower both individuals impacted by ataxia, as well as those healthcare professionals that are caring for those impacted by ataxia. My really wonderful colleague, who's also on the research team, Dr. Celeste Suart, is going to speak after me about many of the patient-directed programs that have been aimed at helping to educate, engage, and empower. But I did want to just mention the over 60 healthcare professionals who have been diligently participating in an ataxia boot camp right down the hall. I think they should be taking their post-test right now, so hopefully they learned a thing or two. Those really are going to be the next generation of folks that are going to replace and fill the very big shoes left by those ataxia experts today. Over 40 of those individuals are fellowships working in movement disorders or genetics or neuromuscular disorders. And we're really, from my count, we're getting about 1/3 of all of those individuals who are in programs like that around the country. So we're really hitting a lot of these folks and trying to kind of direct them maybe slightly away from the Parkinson's field to focus on ataxia research. And finally, in many different ways, every minute of my day is spent in trying to drive drug development. I frequently have one-on-one meetings with many different drug companies, some that you know of, and some that you don't know are working in the ataxia space because they haven't publicly announced it yet. And so I sit at a very privileged position where I get to learn about what's coming down the pipeline before the rest of the community gets to hear it. And so I've told you a little bit about what I do. So before I get started, I wanted to tell you a little bit about why I do it. So on the left here is my grandma, Liz. She started experiencing ataxia symptoms before I was born. And really, through my whole childhood, I really only knew her as being in a wheelchair, and I didn't get to know her very much before, unfortunately, she passed. My father, Bill, who's also sitting in the front row, and you may have heard from earlier today, was also diagnosed with ataxia right as I was making decisions about my future, and which inevitably led me here. And then, of course, there's my sister, Jessica. She spoke just before the break about her family planning, and Jess is one of my favorite people in this entire world, if not my favorite person in this world. Don't tell my 3 siblings, or my other 2 siblings. But this is really why I'm here. This is why I chose to get a PhD in neuroscience. This is why I chose to work with a professor who's specifically working on ataxia drug development. And it's why I worked in a lab for 8 years trying to work on developing therapies for ataxia. And it inevitably, fortunately, brought me here to this stage after 8 years. So I just want to say, I hate ataxia. Like, I really hate ataxia. Can we just, I know I'm not supposed to say that word in front of my nieces, but man, it sucks. It really sucks. I hate that it can take away our grandparents, our parents. I hate that it can cut the careers and lives of especially young individuals short. I hate it. And so that's really why I'm here and why I joined the foundation. And I know we're all ataxia warriors in this room, and we can handle some hard facts. So I'm just going to start this presentation off with just a couple hard facts, because I know you can handle it. Today, there's at least over 300 different types of ataxia-related disorders. I think there's actually probably more than 400 ataxia-related disorders, but I didn't count. And unfortunately, right now today, of all those disorders, only one of them has an approved therapy, and that was the Skyclarys treatment that was approved last year for Friedreich's ataxia. Historically, it takes an average of 15 to 20 years for a drug to move all the way from preclinical development, so those early idea stages, cell testing stages, mouse testing stages, all the way through FDA approval and commercialization. And with all of these facts, it's impossible not to feel overlooked when you have a rare disease. You see clinical trials, you see drugs being developed for all of these other diseases, in some cases rare diseases. And you're wondering, why not me? Why not us? And at the end of the day, no matter how quickly we press things forward, research cannot move fast enough when our needs are so great today. And any promises that I make for you for the future are not going to help you today. And I know that, and I wish I could change that. But these are the hard facts. And at the end of the day, keeping hope can be really hard with all of these things piled up against us. Keeping hope is hard. But what I want to say to you all today is that I am hopeful, and I am not an optimist. I heard about this disease, and I thought, there's no one in this world working on this. I guess I have to do it. I'm not a dreamer, I'm not an optimist, but I am hopeful. I'm a logical scientist, and I am hopeful. And I really do believe that one day there are going to be treatments for these diseases. I think that ataxia is going to be a manageable disease, not a life-shortening disease. I think that individuals who are born with a genetic mutation will one day not have to experience the symptoms associated with that mutation. I really, truly believe that. And so today I want to tell you a couple of the reasons why I'm hopeful, and I've kind of grouped these into 3 categories, and there's many more reasons beyond that, but I'm just going to start with the fact that there is so much more funding going into ataxia research today than ever before, and there are so many shots on goal, some that you are aware of, some that unfortunately I've signed CDAs and can't share with you, but there are so many more shots on goals today than ever before for not just 1 or 2 types of ataxia, but many different forms of ataxia. And then I'm going to finish my presentation today with just one example of how even just a small win in the research world can really have huge and very rapid impacts. So the first reason, why am I hopeful? So today there is more funding being funneled into ataxia research than ever before. And I know this has been said at meetings before, but money buys research and research finds treatments. Money is absolutely essential for any progress to be made in the development of treatments for ataxia. So I was looking at the NIH guidelines for next year, and they're estimating that $7.6 billion will be granted specifically for rare disease research in 2025. This is the most that's ever been granted. It's more than $1 billion over what has ever been granted before. So there has really been a significant shift in funneling money into rare diseases. And then a really huge win for the ataxia community was the addition of hereditary ataxia to this list of diseases that can be funded within a DoD initiative, Department of Defense initiative called the CDMRP. So there's a big pot of money, about $300+ million that's out there for researchers to apply to. And before 2021, that money was not accessible to researchers working in ataxia. And thanks to many in this room, as well as partnered foundations such as FARA, we were able to get first Friedreich's ataxia and then hereditary ataxia added to the list of diseases that can be funded through this initiative. And so just as a reference, the National Ataxia Foundation and FARA, which are above and beyond two of the largest ataxia foundations, we fund a little over $10 million per year. And just through this alone, we've brought more than $25 million into ataxia research over just 2 years. And so we're very excited. I know there was, with this amount of a success, there were many more applications this year going into the CDMRP, and we're really hopeful that we'll have, again, the significant funding being channeled into ataxia research. So next, pharma is investing in ataxia. I know we always talk about, you hear about big bad pharma, but for rare disease, they are absolutely essential, and we need to work with them in every way that we can and help try to create this, make this space, this ataxia space, as attractive as possible, as welcoming as possible, to really encourage them to invest in ataxia research. So as I mentioned, we had our first drug ever approved for an ataxia in February of 2023. That was a drug put forward by Reata Pharmaceuticals. Again, that's Skyclarys for the treatment of Friedreich's ataxia. So following the FDA approval, the company Reata was actually acquired by Biogen, who is one of our sponsors, and you've probably seen their booth right outside. If anyone wants to guess how much Biogen paid to acquire Reata, who at the time only had an FDA approval for this Skyclarys drug, which could potentially be prescribed to about 5,000 or more individuals in the world. Anyone want to guess? Yell it out. How much? $2 billion? $7.3 billion. That is how much a drug company paid to acquire basically one drug for one disease, for one rare disease. That is that is huge. I can't even tell you how huge it is that we were able to prove to the world that developing treatments in ataxias is not only possible, but it's profitable. That is critical. And just as this is just one example of the kind of impact that can have, within a month of Biogen acquiring Reata, there were 2 new companies that had approached NAF and said, we want to join your drug development collaborative. They saw that the successes that were possible, they saw the dollar signs that were possible, and they saw that even just the act of having a drug go all the way from development to approval was possible, and they chose to invest with the National Ataxia Foundation. And just as a note, it wasn't $7.3 billion to join the DDC, it was $100,000 per year for each of those companies. And I mentioned that there's a lot more companies in the background. I can say personally that there was definitely an uptick in the number of companies that we were talking to, and it continues to this day, where there's companies that either I've never heard of or didn't know they're working in the ataxia space, and they're ending up in my inbox and saying, tell me about ataxia. And I know that talking to other researchers and who are going to investing meetings, there's a lot of talk about ataxia in the investing world. So this is a big win for ataxia. All right, so more shots on goal. So what do I mean by that? When I said that the average time from development to approval is 15 to 20 years, we're not starting at year 0. We are well into that development process for many of these potential drugs. And because we've had that strong history, that strong growth, partially funded by NAF grants, can I say, we're at a place where a lot of drugs are finally reaching to the point that they're ready or approaching the point where they can go into clinical trials. And another thing I want to point out is that we're not just talking about your average drugs. Yes, of course, we have some traditional small molecules like the kind of pills that you would take, but we also have some really cool next generation approaches to treat ataxia. We're talking lasers, we're talking DNA drugs, and so I'm just going to spend a little bit of time talking about some of those approaches that have been developed and are going into clinical trials for ataxia. So starting with transcranial magnetic stimulation. Has anyone heard of that here? TMS? I get a lot of emails about TMS, so I thought I should definitely talk about it today. So what TMS is, it's outside of the brain. It's focused electromagnetic pulse to induce an electrical current. So you can see in this picture here, they're able to record where these currents are going, and they're applying it to the outside of the brain. Right now, TMS is FDA approved, but not for ataxia. It's FDA approved for the treatment of depression and OCD. But it's being tested in many, many different neurological disorders, including ataxia. And so the belief is that by stimulating electrical activity in the cerebellum and network and other parts of the brains that are in network with the cerebellum, you could improve cellular processes. So kind of that use it or lose it, but specifically for the cerebellum circuitry. So where are we today with transcranial magnetic stimulation? So I found over 14 clinical studies that have been performed that included over 400 patients with some form of ataxia. What I found in these studies is that they varied widely in the ataxia types included, the treatment approach, and the specific design of the trial. Some of them were a little bit better structured than others, and so it does make it a little bit difficult to interpret and compare results, especially when you're looking at different ataxia types, different readouts. But generally what we have learned from these studies is that this approach appears to be safe, especially when applied in a clinic. Some studies have shown benefit, and some of these studies have shown significant benefit that appears to be sustained. However, there is a broad range of results. In other studies, you'll see that they find no improvement at all in patients. But again, there's just a lot of variability in how these studies are done. So where we are right now is more work is needed. And I know we hear that a lot about these investigational therapies, but this is something that NAF is investing in. So I just wanted to point out one recent grant that we awarded for Dr. Roderick Moss, who is investigating TMS in specifically SCA3. He has studies ongoing, but he does have some preliminary studies published in 2022 and 2023. So we'll keep you abreast of any progress made there, any results that come out of that. There are a couple transcranial magnetic stimulation trials going. I encourage you to go to clinicaltrials.gov and see if there are any trials that you're interested and eligible for participating in. There's also a list of studies that are recruiting patients on the NAF website that I encourage you to check out. All right, next I'm going to talk about gene therapy. Gene therapy. I still have trouble describing to myself what gene therapy is because it's really this all-encompassing term that's sometimes used in different contexts in different ways to describe a whole bunch of approaches. But in this context, I'm going to break it down into gene transfer and gene editing. So gene therapy, at its base, describes multiple approaches or technologies that aim to treat genetic disorders at the DNA level. So you're either supplying the cell with DNA through gene transfer approaches, which typically involve some kind of virus to deliver the sections of DNA into cells, or gene editing. And many of you probably have heard the word CRISPR, which is actually an acronym that I'm not going to say because I'm going to mess it up. But CRISPR won a Nobel Prize in 2020 for some of the investigators that were really pivotal in developing this approach. And so when I'm talking about CRISPR, I'm talking about going in and editing DNA within a cell or organism's genome. Which, don't we all just wish we could fix our DNA? So I'm going to start with gene transfer because that certainly has progressed further in the ataxia space. So gene transfer is the delivery of genes into cells. And this is particularly promising in diseases where a mutation leads to not enough of a protein or not enough of a functional protein. And those are very often recessive diseases. And so a lot of the progress has been made in Friedreich's ataxia, which is a recessive disease where typically FA patients have only one copy of a functional FXN gene. And so the aim of a gene transfer approach is to compensate this lack of functional frataxin by supplying cells with the brain or the heart with a functional frataxin. And again, this is typically done by using a viral vector. So there have been multiple trials that have begun in Friedreich's ataxia using a gene transfer approach. And excitingly, one of the first gene transfer approaches went into clinical trials that target the heart. I believe that started last year. So there are 2 ongoing gene transfer trials that I believe are recruiting at this time to assess gene transfer approaches in cardiomyopathy. So what about gene editing via CRISPR? It sounds like the dream, to be able to go into every cell and make corrections and just get rid of the thing, no more ataxia. And I would say that where we are right now with CRISPR is that we've gotten really good at selectively modifying the DNA of individual cells. And in some cases, we have been able to use this approach to develop really, really effective treatments for some diseases. So the first CRISPR-based therapy was FDA approved in December of 2023 for the treatment of sickle cell disease. So the real question is, can CRISPR-based gene editing approaches be used to develop treatments for ataxia? So what is CRISPR good at today? It's really, really good at rapidly creating new disease model systems. And I know that doesn't sound exciting. That doesn't sound like a treatment. That doesn't sound like a cure. But when you have a rare disease, previously it would take maybe multiple years, if not 5+ years, to develop a mouse model that can model that disease and then can be used to study, understand the disease, test treatments, develop treatments. CRISPR has enabled that timeline to go down to about a year. So a new gene is discovered. Within a year, scientists are typically able to start studying that in a living organism and really start to understand and parse out what's going on and how we might be able to fix it. Because it's really good at creating these model systems, it's really good at testing potential therapies and also investigating disease model systems. So if you have, for example, an ataxia mouse model and you're like, I think this other gene might be playing a big role in that disease, you can quickly make changes to that specific gene and see what happens, see if your hypothesis is correct. So CRISPR is actively being used to create model systems and to test theories in labs throughout the world. And I think just about every major university now has cores, facilities that specifically use CRISPR to create mouse models and other cellular models of disease. And just for an example, I selected just a couple of the many different grants that use CRISPR in their labs that NAF has awarded over the last couple of years. And then finally, it's proven to be very effective for therapies for blood disorders and for some cancers. And for these diseases, the DNA editing is typically being done outside of the body, or it's being done in a very targeted, isolated tissue. And the reason that is that CRISPR makes a lot of mistakes. So it's not a great editor, and sometimes it has the risk of making incomplete or incorrect DNA changes. And so it's much better if you can do this outside of the body and then see what happens, make sure that you haven't made too many mistakes, and then before putting it back into the body. It can also stimulate the immune system and activate an immune response. And then finally, we have the problem of how do we get all of these CRISPR components into every cell in the affected tissue, and then hope that they're all editing correctly and not making really detrimental mistakes that might cause more harm than good. So where we are right now is that CRISPR technologies are and will continue to improve at a rapid rate. I think there's already a new technology that might kind of overtake CRISPR within the next couple of years. So I have no doubt that CRISPR will continue to progress and move into neurological disorders. And in fact, it has progressed to human clinical trials for some neurological disorders, such as Rett syndrome. But right now, CRISPR is not actively being used in clinical trials for ataxia, but is very much being used in drug development for ataxia. And what about nucleotide-based therapeutics? So before, with the gene transfer and the gene editing, I was talking about either delivering DNA or targeting specifically the DNA. But there are ways of changing the expression of mutated genes that don't directly modify or target the DNA, but instead target the RNA. And I'm not going to bore you with an in-depth discussion of that, but what I can say is that what we've actually done is found that we can use short pieces of modified molecules that look a lot like our DNA or RNA, and those can be used to turn, for example, if you have a gene mutation, and that gene mutation is a light. We can't unplug the light using this approach, but we could potentially turn it on or off using these nucleotide-based therapeutics. And I'll just mention that much of my graduate work was focused on assessing antisense oligonucleotides, one of the nucleotide-based approaches in SCA3 mouse model systems. So I think those ASOs, or antisense oligonucleotides, are really set a good example for what we might expect over the next coming years in ataxia drug development. Last year was a really exciting year for SCA3. There were 2 first-in-kind clinical trials that were launched last year that tested different ASO therapies targeting SCAs. In one case, it was just SCA3. Another case, it was SCA1 and SCA3, as well as Huntington's disease. So one of those trials was unfortunately halted in April of 2023, and this was likely due to non-clinical safety data. However, the other clinical trial, which was Vico Therapeutics phase 1/2A for their ASO VO659, is still ongoing in European patients with SCA1 and SCA3. That trial isn't expected to end until, I think, early next year. And we're in a stage where no news is really good news. We would only hear results of that trial if there was some issue that might cause it to be paused or stopped. So I'm very excited to hear where that trial might go, and when it might move into the next phase of clinical development and potentially into North American sites over here. So one thing I want you to take away from this example of ASO therapy for SCA3 is that drug development is volatile. You're going to see successes, but you're also going to see a lot of failures. And that's what we should expect. And rarely drug development is going to progress in a straight line. So behind the scenes, many more companies are investing in nucleotide-based therapeutic programs for a wide range of genetic ataxias. I mentioned SCA3, but there are antisense and other approaches being developed for many different forms of genetic ataxias. And then one other takeaway is just that we gain a wealth of information from every single failed clinical trial. So I don't really want you to think of a failed clinical trial. Anytime we can move a drug into the clinical trial stage, regardless of the success or failure, we learn so much from that trial. And every failure, every success makes the likelihood of a successful trial in the next potential treatment, the next promising treatment, even more likely. And so today, nucleotide-based therapeutics remain a promising and actively explored approach for treating genetic ataxias, despite some of the setbacks that were seen last year. All right, so I talked about more funding going into the ataxia space, and I talked about more shots on goal with this kind of new age arsenal of weapons that we have to attack ataxia. And so I want to talk a little bit about one specific story of how small wins can have a huge impact. And that's with the SCA27B story, which you heard a little bit already this week. But in fall of 2022, a new genetic cause of ataxia was discovered. And that disease that inevitably was discovered was called SCA27B. And in fact, a couple of the researchers who were involved in identifying that gene are here today, and two of them will be receiving an award later for their contributions to the field of ataxia. All right, so that was fall of 2022. So where are we a year and a half later? There's been a rapid development of a DNA test that has enabled more than 1,200 patients to be diagnosed with SCA27B. 1,200 patients. These are from families who previously did not have a name for their disease. They did not, they didn't have a story for their disease. They weren't able to diagnose themselves, find out the risks for their children. And today they were able to have a diagnosis through both the progress of identifying the gene and then also through the major progress that has been made in development of genetic testing approaches. At a quick glance, more than 25 research studies have been published on this newly discovered gene. And these studies have found that SCA27B may in fact be the most common form of dominantly inherited ataxia. Only time will tell, but it is extremely common, especially in specific regions around the world. And a couple of these studies found that symptoms of this disease appear to improve following treatment with a specific drug that's FDA approved for multiple sclerosis. And so within a year and a half, there was at least one drug company who saw these results and thought that this is something that should be tested and tried and put through the evaluation of a clinical trial. And so Ataxia Pharmaceuticals, who is one of the sponsors of this meeting today, is in pre-IND discussions with the FDA with the hopes to try to run a clinical trial starting as early as 2025. So when I talked about that 15 to 20 year process, we're looking at a clinical trial after potentially 2 years after a new ataxia type was discovered. I just think that's incredible, and I think that this is not going to be a unique story, but is going to be replicated across multiple diseases, just because so much of the groundwork has already been laid and all the technologies are out there for us to really make rapid advancements in drug development for different forms of ataxia. And of course, I talked about the impact on patients, but this and how it gave answers to families, and one of those families was Bill Nye's family, and that brought him to the National Ataxia Foundation, and who knows where that's going to go from here, but we really hope that this is going to help get the word out about ataxia and just make it a little bit easier to not have to explain ourselves out in the world and to get funding to support these rare diseases. All right, so why am I hopeful? I talked a little bit about how we are, excuse me, have brought more funding into ataxia research. We have many more shots on goal. We've started that 15 to 20 year clock on many different therapeutics and are approaching treatments for many different diseases. And even just a small win can have really huge impacts on many, many people and on an entire field. And I know this is going to sound a little bit cheesy, but it's really all of you that make me very hopeful. And all of these different steps, major steps, minor steps that have been made in the development of treatments for ataxia were only possible because individuals like yourselves participated in research or advocated for the community. And really, I know at least I won't, and I know all of you won't, we won't accept failure. and our voices are going to be heard whether they like it or not. And so I just wanted to highlight a couple ways that patients have been pivotal in the progress that's been made. So every year we have our United Against Ataxia Hill Day. It was specifically through this, initiative that we were able to get hereditary ataxia added to the CDMRP. So thank you everyone who has participated in previous Hill Days, and I hope you'll all continue to do that in the future. You're able to do it virtually from your home, and so I think it's just a really great option for ataxia patients to make sure their voices are heard in Washington. We have a National Ataxia Awareness Day, if you weren't aware. It's September 25th, so that is always right around the time that we do our Hill Day each year. Last year, we had the opportunity for the first time to lead a patient-led listening session. So with many of the drugs that we're developing and moving forward in clinical development for SCA3, we thought it was a really good time to teach the FDA a little bit about this disease. And so we gathered, I believe it was 6 or 7 patients who gave talks to the FDA and taught them about SCA3. And from that, we discovered, we really both taught the FDA about the SCA3 and ourselves learned a lot about the specific symptoms that impact patients. And it has helped mold how we run our natural history study and could impact the way we run clinical trials in the future. I know many of you banded together to participate in the community response following the refusal to review the troriluzole NDA last year. We were able to get 3,137 signatures. The troriluzole NDA is still being reviewed by the FDA, and Biohaven, who owns the troriluzole asset, is still in discussions with the FDA, so we'll keep you all abreast of any changes there. And then I'll just mention that it's not just us, it's not just FARA. There are other global and federal programs that are focusing on ataxia, and so I'll just mention that the Critical Path to Therapeutics for the Ataxias was started, I believe, in 2022, and they are focusing on preparing the ataxia field for future clinical trials. And then the Ataxia Global Initiative, which really was born at a AAC meeting several years ago, has grown into a worldwide consortium of researchers and clinicians that are working on developing treatments for ataxia. So these are just a few of the reasons why I am hopeful, and I hope you are too. So with that, I just want to say thank you, and thank you for staying through the whole weekend to listen to me.
Thank you very much. I'd like just to thank you for your effort in promoting research by DoD, because last week, before I came here, I submitted the first grant proposal to the DoD for ataxia research, for an idea that we have. So I'm a researcher for how to treat the intention tremor. And I didn't know about all this amazing effort behind NAF, and it's really, this is my first time here, so it's amazing to see all the people behind that. And then I have another question. We also got a ARPA-H grant for repurposing drugs. And you talk about rare diseases. The goal is to use AI to mine all the molecular profiles of the 3,000 FDA-approved drugs against all the 7,000 rare diseases and their molecular profiles. And I think I like to push that towards ataxia, if possible, and for that I need help. I need to know who to talk to, and this is my first time seeing this community. So if you have ideas how to do the next step.
I would email research@ataxia.org, and we'll have a chat pretty soon.
Yeah, okay. Thank you very much.
Thank you. Oh, sorry. Thank you for your very informative talk. It's great to hear all the advances. I have a question about ASOs. And is the thought that they would prevent the progression of a disease, but once for example, in the case of SCAs, once damage to the cerebellum is done, they could not reverse that, but they could prevent that progression from happening?
I think it may be a bit too early to say. I think primarily it would be in slowing disease, or slowing progression of the disease. But these haven't progressed far into clinical trials, so we'll just have to wait and see.
Dr. Moore, thank you for your informative talk. I was wondering about last year we heard about a number of viral vectors that use RNA inhibition to knock down the gene products of autosomal dominant diseases, and I was wondering, once we have the proof of concept clinically of a treatment that will take down diseases caused by a triplet codon expansion for one specific ataxia, will the same regulatory hoops have to be jumped through for other similar ataxias that operate by the same model?
So I would say any new compound that has never gone into humans is always going to have to go through a phase 1 dose escalation study. So we're always going to have to do that early safety approach. We certainly are going to learn a lot from the first drug that's approved that may make improvements in how we design trials, how we dose, how we assess changes. But there's always going to have to be that phase 1 dose study. Mm-hmm.
Thank you. When you mentioned about the 2 clinical trials for SCA3 last year, and one was halted due to not enough clinical data, did you mean not enough subjects?
No, by no means no. There was definitely enough SCA3 patients out the door who were willing to do that. No, the press release said non-clinical safety data. And so it didn't have anything to do with what was happening in the patients. That would be clinical safety data. So this was some safety data that was not occurring within the clinical trial, but did cause concerns for continuation of the trial. And if you're confused, that totally makes sense. Many times these are vague by nature, yeah, I don't know the exact reason why it was stopped, but it was non-clinical safety concerns. Mm-hmm.
Thank you, Lauren, for a nice talk. It really was helpful. I want to put something in perspective. In the early 70s, our first grant was given to Bob Currier for $5,000. And that led to the discovery of the Ataxia SCA1 gene in 1993. That was only 20 years. And I would think now, the other thing is that Huntington's disease is a CAG repeat disorder, and they use tetrabenazine with success, to some success. Has that been tried in any of the ataxias?
I'm not actually certain off the top of my head, but I'll certainly be looking up into that after this meeting. One of the ASO trials that I mentioned, the part of the gene that the ASO is targeting is this CAG repeat. And so that's why they're able to use the same exact drug in testing in 3 different CAG repeat diseases, that being SCA1, SCA3, and HD. So at least in a gene targeting approach, depending on which part of the gene you're targeting, you might be able to use the same approach for multiple diseases, but I'm not certain about the compound you mentioned, sorry.
I haven't heard much about the CORDS opportunity and registry. Is that still a place where someone would go if they want to be involved in research?
Yes, it is. So the CORDS registry is basically a contact list for individuals who have ataxia and are interested in having the possibility of participating in research. So there are a number of different companies that will approach, companies or researchers who will approach NAF and say, I want to recruit for this study, and we will post that on our website. But there are also many researchers who go straight to CORDS and maybe don't come to NAF for research studies. So, if there are multiple multiple registries that you could join, I would highly recommend you to do that. Especially if you're interested in research. So if you go to the CORDS website and join their registry, you'll be contacted only if there are studies that you might be eligible for. And it also really helps. One of the number one questions that I get asked by drug companies is how many people are there in the US with disease X, disease Y? And right now, we really don't have a good number to tell them. And so registries, NAF membership, and other programs like that really help us to get a better idea of how many patients there are in the US and really help encourage drug development. If they know both clinical trials are possible and commercialization is possible for specific diseases.
Thanks, Lauren. We're up on time now. That was a great presentation.
Thank you.